Everyone. So my name is Kate Shulgina. I go by Kate. And I'm currently a postdoc at UC Berkeley. But today I'll be talking about my PhD work, which was done with Sean Eddy at Harvard, where I analyze microbial genomes for alternative genetic codes. So the genetic code is how codons in mRNA get translated as amino acids in a protein sequence. And this same genetic code is used by almost all of life. It's so conserved in part because any changes to it would probably affect how most proteins are translated. But exceptions do exist. For example, our own mitochondria use a different genetic code. Here, several codons are different. So this stop codon UGA is now a tryptophan codon. Isoleucine codon AUA is a methionine. And these two arginine codons are stops. So when I see alternative genetic codes like this one, a lot of questions come to mind. Firstly, how do you even evolve a change to the meaning of a codon without disrupting the production of proteins? Does a codon first need to drop to zero usage, or could there be an intermediate period where it's translated ambiguously? Secondly, what sort of mutations to tRNAs and other translational components enable these reassignments? And could they bias what sort of changes are easier to evolve than others? And in the past few decades, even more genetic codes have been found in various bacteria, eukaryotes, many more in mitochondrial genomes, and even in viruses, to the point where this figure from 2016 is starting to get a bit out of date. As we're sequencing more of the microbial diversity, more and more alternative genetic codes continue to be found. However, one challenge is that we lack a computational method that's widely used for predicting the genetic code from a genome. So most of these examples here were found incidentally, not through a systematic screen. So it's hard to know if the patterns we observe here in, say, what reassignments are more common reflect real biology or just observation bias. And furthermore, if we fail to recognize that a genome, a newly sequenced genome, uses an alternative genetic code, then that will cause errors in protein prediction and in downstream public databases. And so our approach has been to perform the first systematic screen for new genetic codes in most sequenced organisms. And from this, we hope to learn more about how and why the genetic code evolves. So to do this, we developed a computational tool called CODETA. And CODETA takes a nucleotide sequence like a genome as input, and then for each of the 64 codons, predicts the most likely amino acid meaning, or if the codon has no amino acid meaning, like a stop codon, or if there's just not enough information, then that codon is left uninferred, represented by a question mark. So now I'm going to walk through how CODETA works by using the genome sequence of Mycoplasma bacteria as an example. And mycoplasmas have reassigned the UGA stop codon to tryptophan. So we're going to see if we can predict that. So we start off with the genome sequence. And the first step is to create a 6-frame translation using the standard genetic code. So here I'm just showing one of those 6 frames. And you'll notice that canonical stop codons like UGA are translated as X, which allows for the possibility of them being sense codons. Then we align this preliminary translation to protein domain models from the PFAM database. So here's just this PAD-PORF domain aligned to this bit of sequence. And these PFAM models contain these position-specific emission probabilities represented by this heat map, which indicate what amino acid is preferred at each domain position. So the PFAM database consists of over 19,000 models of protein domain models from across the tree of life. So no matter what organism you're analyzing, you're likely to get many domain hits. So then to predict the meaning of a single codon like UGA, we take all the PFAM positions that have aligned to UGAs across the whole genome. And then we use a statistical model to ask what amino acid translation of UGA is most likely, given that this is the collection of aligned positions. So for example, if UGA were a tryptophan codon, our model would expect to see many columns that are conserved for tryptophan. And indeed, that's the case here. And if we repeat this process for the remaining 63 codons, we could predict the entire genetic code. And we did some validation of our method on synthetic sequences, which I'm not going to show, but they gave us the confidence to proceed to looking at real biological data sets. So the first clade we looked at were the yeasts, and yeasts are interesting because depending on the species, they translate the CUG codon as either leucine, alanine, or serine. And we analyzed 462 yeast species, and you can see that the codetta prediction was mostly consistent with what we'd expect. One clade really stood out to us, highlighted in gray here, which at the time wasn't known how it translated CUG. And codetta predicted for some species it would be serine and for others uninferred. So then we looked at the tRNA genes to decode CUG. And most of these species actually contain two different tRNAs, one that looks like a leucine and one that looks like a serine. So if CUG is being decoded ambiguously, it would explain why codetta didn't want to select a singular amino acid model in many of these cases. Then we wanted to show that these tRNAs are functional, so we focused on a single species, Saccharomycopsis melonga, and performed northern blotting. So these top plots show that these tRNAs are both expressed, with the left two lanes as controls for probe hybridization. And then we did acid urea page northern blotting on the bottom, which shows that these tRNAs are predominantly amino acylated, indicating that they're probably functional. So in summary, looking at the yeast showed that codetta can not only predict normal codon reassignments, but also flag unusual cases like ambiguous translation. And in the time since these experiments, other groups have validated the ambiguous translation by proteomic mass spec. Then we moved on to a large-scale analysis of 250,000 bacterial and archaeal genomes from GenBank. And to simplify, I'm showing one genome per species. So that's about 48,000 species, most of them bacteria. And for the vast majority of them, codetta predicted no deviation from the standard genetic code. We also found all of the previously known alternative genetic codes. So these are mostly mycoplasmas in bacteria. We had one common false positive crop up in genomes with high GC content that we believe is due to non-synonymous substitutions from lysine to arginine codons. And then for about 300 species, we had some change from the standard genetic code. So this could be a mixture of real new genetic codes or errors. So then we had to look through each of these candidates for additional computational support, for example, tRNAs. And we ended up with 5 clades using new reassignments. This represents about maybe 30, 40 species total. And interestingly, all of these new reassignments affected arginine codons, representing the first sense codon changes in bacteria. So now I'm going to go into some of these reassignments in a little more detail to see what we can learn about how they evolved. So in this clade of uncultivated enterosoma, we predicted that AGG, an arginine codon, is now methionine. And the outgroups use the standard arginine decoding. As a sanity check, we looked at multiple sequence alignments of conserved single copy genes like uridyl kinase. And you can see that in the reassigned clade, AGGs, indicated by these alphas, occur at positions that are predominantly conserved for methionine, consistent with the codetta prediction. Then we looked at the codon usage of AGG compared to the canonical methionine codon AUG. And you can see that in this clade, AGG is actually the dominant methionine codon. So this tells us this is a mature codon reassignment. Not only has AGG been removed from arginine positions, but it's also synonymously substituted into many methionine codons. Then we looked for the tRNA that could decode AGG, so that would have the CCU anticodon. And the reassigned clade has a tRNA that has features consistent with methionine identity, highlighted in blue, which are recognized by the methionyl tRNA synthetase. And the outgroup tRNA looks like an arginine tRNA. And when we looked at the genomic context of these two tRNAs, it turns out they're flanked by the same two surrounding genes in both the reassigned clade and in the outgroup. So this tells us that these two tRNAs were likely derived from the same ancestral tRNA gene, and the ability to decode AGG as methionine evolved due to mutations that changed which synthetase is recognizing this tRNA. And this was pretty surprising to me because if you had asked me what's the easiest way to get a codon reassignment, I would have said just duplicate a methionine tRNA and mutate the anticodon. But here it seems like a different path was taken. And then for the last 4 reassignments, they all affected the CGA and CGG codons. And what's interesting is that all of these 4 clades have pretty low genomic GC content, around 30%. And here's B subtilis just for reference. And if we look at the codon usage per 10,000 of the 6 standard arginine codons, so that's the reassigned codons and the unaffected ones, you can see that the GC-rich codons like CGC, CGA, CGG have really low usage, while the AT-rich AGA codon is favored. And this is in contrast to the more even usage of arginine codons and subtilis. So what we think happened is that this low GC content might have driven these codons to low usage, which then facilitated the reassignment by minimizing the potential detrimental impact on protein sequences. So in summary, we developed CODETA, a computational tool for predicting the genetic code, and I hope it will be more broadly used by the community, not only to find new genetic codes, but just to confirm that you're assuming the correct genetic code when you sequence a new microbe. Then we confirmed the CUG reassignments in yeast, and in a large-scale screen, found 5 new codon reassignments in bacteria. And with that, I would like to thank my PhD advisor, Sean Eddy, and everyone who supported me through this project. And as I mentioned, I'm currently a postdoc in Jamie Cate's lab at UC Berkeley, where I'm working on ribosome structural biology. Happy to take any questions. Nice.
That looks like a lot of fun. Is every PFAM equally informative for this process?
Every what?
Every PFAM.
Yes. I guess if they're more conserved, that's more informative. Some positions just aren't conserved, so they don't tell you much. But we use all of the positions in the predictions.
That was really interesting. You didn't talk about any stop codon reassignments. Is that because CODETA doesn't assess them as much, or is it because you didn't find any?
We didn't find any. CODETA can find stop to sense and sense to sense reassignments. We don't explicitly predict stop codons, so we can't find sense to stop changes. But yeah, I guess the stop codon reassignments are easier to find without a method like this, because if you sequence a genome and you notice there's in-frame stops everywhere in your ORFs, you're gonna realize something's up, whereas a sense codon reassignment is a much more subtle signature.
Thank you. So in these organisms where you have reassigned the putative translation table, do you see a increase in coding density with the new table as compared with the sort of default assumption?
You mean like with a gene predictor? I haven't extensively looked, but since these are sense codon reassignments, I wouldn't expect there to be a difference.
Do you know it may be such cases when one codon codes for 2 amino acids in the same genome? So yeah, if you sometimes you see that in the same genome, when you do multiple sequence alignment, the cis codon it's more aligned with some amino acids, and in another genes, it's with another. So is it possible that the same species may have tRNA which have the same anticodon but different amino acids?
Well, that's kind of what's going on in the yeast example. And so the yeast that have 2 tRNAs to read CUG, the leucine and the serine, there's evidence suggesting that these tRNAs are actually expressed in a condition-specific manner. And so the CUG is occurring in conserved serine positions in some subset of genes and conserved leucine positions in another subset. Yeah.
Okay, so there are 2 questions from Zoom participants. Brian Bushnell asked, how long does it take to run on a typical bacteria-sized genome?
If you run just like a laptop computer, maybe 10 to 15 minutes. Obviously, when I did my large-scale screen, I used parallelize it over a large computing cluster. If you're going to analyze eukaryotic genomes, I suggest parallelizing as well. So the time-intensive step is the alignment of the PFAM models to the translations of the genome So it just scales with the size of the genome and yeah, essentially.
All right, the second question is from David Donegan. Have you used CODETA to evaluate DNA viruses? Many encode multiple tRNAs.
I haven't, but I encourage anyone who's interested to do so.